I'm taking the summer off writing a book about consciousness in the brain, and this is kind of what the book is oriented on. And I gave Gautam 3 titles, all of which are potential candidates for the book title, and he picked this one because it's the safest. Now, the one I prefer is this one, Tuning the Brain, How Neuroscience Got It Wrong and Quantum Vibrations Make You Feel All Right. And he said, no, the neuroscientists won't come. They'll be offended, but you're here anyway already. So I think neuroscience has gotten it wrong in terms of understanding how the brain produces consciousness in a lot of things. I mean, obviously, we know an enormous amount of details. We know a lot. But how does the brain really work? I don't think we really know. So part of the dogma of how the brain works comes from, or is summarized in a paper by Crick and Koch in 2003, A Framework for Consciousness. And this kind of, consciousness had been kind of buried, wasn't really talked about. And Crick and some other prominent people, Penrose, Edelman, came out in the late 1980s and started talking about consciousness. And anyway, Crick teamed up with Christoph Koch, a very notable neuroscientist, and they came up with a framework for consciousness. Kind of set the frame or the stage for what we should be looking for in terms of understanding consciousness. And they gave 10 rules, kind of like the 10 Commandments. I'm joking, obviously, but the major point they made was that the cortex is the primary site of consciousness, which is probably true. The front of the brain looks at the back of the brain, which is also sort of true. We have zombie modes of non-conscious autopilot activities that go on without us being conscious, which is true. Consciousness involves coalitions or assemblies of neurons, which is basically true. It goes back to Hebbian assemblies. There are explicit representations, for example, in V1. I think that's true. Higher levels get activated first, and then consciousness happens later, and that's probably also true. There's modulation of firings, which is probably true, although I don't think firings are what mediate consciousness, and I'll get to that point. Snapshot moments, that is, consciousness is a sequence of discrete events, and I think that's true, and that's probably the most profound thing that came out of this, because everything else was kind of already known. There's attention and binding are problems, and there's a penumbra of non-conscious that kind of tied it to William James and so forth. But always talking about firings. Now, since that time, since Francis Crick passed away, Christoph has aligned himself with Giulio Tononi, so I'd add 3 more commandments, if you will, that consciousness involves integration of information and complexity, that phi terms... the Tononi terms phi, complexity, and lately Christoph has resorted to panpsychism. I think he realized that the computational approach didn't work and explain consciousness, and now becomes somewhat of a panpsychist in his recent book, Confessions of a Romantic Reductionist. I'm not picking on Christoph. I like him a lot. I just think he kind of symbolizes where we've gone, and he is one of the leaders. And I think we're missing the boat. I think we're barking up a lot of wrong trees. Okay, so the dogma as it stands, if you distill all that and take where we are now, I think this is probably what people talk about or is taught. Everything's based on the Hodgkin-Huxley neuron, which I'll get to in a second. That it's integrate, and fire. So inputs, integrate to a threshold, then fire. And people look at the firings, the spikes, and their synaptic transmissions as fundamental information bits, despite the fact that local field potentials in EEG come from dendrites and soma, not from spikes. And local field EEG is the best marker we have of correlative consciousness, despite the fact that the emphasis is on spikes, because they're easy to measure. You can put them on sound and you hear that kind of stuff, and it's really cool. But consciousness may have already happened by the time the neuron fires. I think it happens, which I'll get to, in the dendrites and soma. Everything is based on the neuronal surface membrane potentials, mediating signaling, graded potentials in dendrites and soma, and the all or none action potential, EPSPs, IPSPs, and spikes. That is everything in terms of information processing, in terms of real-time cognition and consciousness in the dogma. Psychoactive drugs, anything that affects the brain, mood, consciousness, act exclusively at postsynaptic membrane receptors or channels. 5-HT, GABA receptors, dopamine, opiate, NMDA, et cetera, et cetera. Everything, all the drug studies are aimed at what receptors being activated. And I think they do bind to receptors, but they also bind inside the cell, as I'll show you. And that the neuronal interior, including the cytoskeletal microtubules, provide only structural, logistical support. They're skeletal structures and transport structures. Okay, so that's kind of the summary of the dogma where we are right now. Now, what does this not explain? What neuroscience dogma cannot explain? Well, consciousness, awareness, phenomenal experience, the hard problem. Okay, maybe I'm being picky because you can say, well, nobody can explain that. But I'm not sure that's true. I think that, barking up the wrong tree, for sure we can't explain it. Memory. Synaptic plasticity supposedly mediates memory, but membrane proteins which mediate synaptic plasticity are transient, hours to days, and they're gone. They're recycled. Yet memories can last lifetimes. Real-time conscious control, as measurable brain activity correlating with consciousness apparently occurs too late. So must be epiphenomenal and illusory, and I think that's not necessarily true. Single-cell organisms with no synapses, just one organism, swim, learn, and have sex without synaptic connections using their microtubules. If a paramecium is that clever, are neurons just on-off states, 1 or 0? Mechanism of action for anesthetics and psychoactive drugs. People say this receptor, that receptor, but we don't really know. Treatments for Alzheimer's disease, traumatic brain injury, etc. We don't really have any good treatments. Every paper comes out and says this is going to help treat Alzheimer's, and of course they never do. And the possibility for non-locality, near-death experience, out-of-body experience, telepathy, these things are just say, no, that's impossible because the brain is a computer, blah, blah, blah. Well, how do we really know? There's certainly a lot of reports and this sort of thing of consciousness leaving the body. And the same, I don't claim any proof of any of this, but there's certainly a lot of reports. And the only reason we say it's impossible because we have this dogmatic approach to the brain, which doesn't really explain anything important that I can tell, important in terms of consciousness. So we can't really say that there aren't these experiences. Okay, so the basis for the dogma is based on the integrate and fire Hodgkin-Huxley neuron, where inputs come into dendrites and soma, the cell body, from all these synapses all over here. It's integrated to a threshold, which when threshold is met, fires. And the signaling is by ions traversing, causing signals along. And then that goes all or none all the way to the next layer of synapses, with about an 85% probability of releasing neurotransmitters to the next synapse. So integrate, fire, integrate, fire, integrate, fire. That's the basic model that we have for Hodgkin-Huxley neurons. You put all these together, and you have the brain as a computer. If you take enough integrate and fire neurons, put them together, you can make a computer out of something that looks like a computational system. And that's the model. That's what everybody's bought into. The brain is a computer. Neurons, firings are bits. Integrate and fire neurons. However, a couple problems. Number one, neurons aren't really Hodgkin-Huxley integrate and fire neurons completely. So, for example, this is from a paper by Nandorf et al. In 2006 in Nature, where here is what would be predicted by the Hodgkin-Huxley neuron, with the red being spikes and the gray being activity in the soma and dendrites. And basically, when it reaches a threshold about here, there's a very narrow threshold, and the spikes occur at an angle. And the reason is supposedly that they have to propagate along these ion channels in the axon initiation segment. That's why they're slanted. But there's a very narrow threshold. So when you reach the threshold, you fire, and that's a 1 as opposed to a 0. That's what Hodgkin-Huxley behavior is predicted to be. However, Nandorf et al. Put electrodes in cortical neurons in awake animals, and what they found was different. What they found was that the threshold was quite variable and also upright. So all the spikes happened vertically at the same time, which means that these ion channels must have opened simultaneously, some kind of coherent opening. That's for the verticality. But most significantly, we have this very wide variability. Something other than the membrane potential is triggering the neuron to spike. There's some X factor. Yeah?
What's on the y-axis here?
Voltage, I think. Yeah, microvolts. These are the spikes, and this is the EPSP, IPSPs.
It's the time along the bottom, right?
This is memory potential. Yeah. That's a good question. I'm not sure, actually.
With the grouping of the vertical lines, that corresponds to cotemporal firing. Is that what you're saying of a bunch of pairs?
It happens at different voltages. It can happen down here at whatever that is, 60. It can happen at 54. And it varies from spike to spike. That was the point of the paper. There's some additional factor, in addition to memory potential, that triggers a spike or not. Something coming from where? From, I would say, inside the neuron. That doesn't show in the memory potential. So at a given memory potential, the neuron should fire. This shows that at a given memory potential, it may or may not fire, depending on some other factor. There's a lot more variability. And they also did it in slice, and they did it in simulation, and they didn't see this variability. So it had something to do with this neuron being in an awake animal.
So the Y would then be counts?
Might be current.
I think, yeah, these are individual spikes. Yeah, I'm not sure, actually. There's the pay. We can look it up, or I can look it up.
Can I ask you, what were you referring to when you said those channels opened at the same time?
Well, the Hodgkin-Huxley, the reason these are supposed to, it's slanted, is that this opens and this opens and this opens and this opens, so it's gradual. But what actually happens is, it looks like they're all opening, or a bunch of them are opening at the same time. Some kind of quantum coherence, maybe, which has been suggested for that. All right, so there's some additional factor. So if we put in a schematic, basically what it says, if we have integration over time versus memory potential, there's a very narrow temporal window and a very narrow voltage window for firing. But what is actually observed is that there's a very wide temporal variability and a very wide voltage variability that influences when there's firing. And what I'm suggesting is this is where consciousness comes in. When I say bing, that supposedly means consciousness. So this would be the strategic place for consciousness to come in and affect firing. Otherwise, we're in a deterministic loop. Everything is algorithmic, and well, besides no free will, we'd be automata. But if there's variability, this is where it would come in and strategically and logically affect behavior and activity by altering the threshold from somewhere. I'm saying inside the cell. Okay, so a couple of preliminary conclusions. Consciousness occurs in end integration, and with that I agree with Tononi, except then they bring in spikes, which integration has already happened. Consciousness causes deviation in Hodgkin-Huxley neuronal behavior. I think that's where we should be looking for consciousness. Where and how? Okay, so let's just consider sensory inputs, which go, visual or whatever, go to thalamus. And we know that there's basically 3 waves of activity, at least for visual consciousness. The first wave, primary, goes to V1, as Crick and Koch said, which is not conscious but may have a representation. And then there's a secondary wave, feedback, which goes to the front of the brain, prefrontal cortex or some other area. And then there's a third wave that goes from there somewhere else, various places, and that's conscious. We know that from a number of types of studies. So in philosophy, this is similar to the higher order thought theory, Rosenthal and Genera, that you need some kind of executive cortex feeding back to cause consciousness to happen. Now, we were just talking earlier about that study by Ralphie Malik where they watched the movie and there wasn't any frontal activity. It was all back. So this is for this particular type of consciousness. Maybe you don't need this for everything, but for this particular form, it seems to work. Another reason it seems to work is that this third wave, frontal parietal feedback, Victor Lame has done. He's the one who suggested it's the recurrence or the. I'm sorry. This is feed forward, and then this is feedback. So the feedback, feed forward is not conscious. The feedback or the third wave is what correlates with consciousness, and that's what Victor Lame and others have said. Now, also, George Mashur, who spoke at the Tucson conference, did a very interesting study. It turns out that all anesthetics, whether it's gas anesthetics, ketamine, propofol, the 3 basic types, the gases, propofol, and ketamine, all act only on this third wave. It doesn't affect this. We get nice evoked potentials under anesthesia. It doesn't affect the feedback, the feed forward. It does inhibit this feedback, this third wave. And why that is really strange, because the neurotransmitters, the receptors, they're all the same for all three waves. But something specific about the third wave that seems to be relevant to consciousness. So the third wave, what happens when it gets to cortex? And there's also three waves. The primary inputs come to layer 4. And they travel along layer 4. And layer 4 sends inputs. This is supposed to be green, yellow, and red. It kind of looks the same. From 4 it goes to 1, 2, and 6. 1, 2, and 6. And then from 1, 2, and 6, we get this third wave that goes to the pyramidal neurons in layer 5. The giant pyramidal neurons in layer 5, whose apical dendrites go to the cortical surface and give rise to EEG. So we have this third wave also at the level of cortex. And this seems to be where consciousness is most likely to happen. I'm not saying it happens only in pyramidal cells, but if it happens anywhere in the brain, and it must, it should involve pyramidal neurons. It could involve others. Now, if we look inside a pyramidal neuron, which is the origin of EEG, we see microtubules. And here's the basilar dendrites, which are much bigger and may go out, and apical dendrites go to the surface. And here's the axon going down here. And we see that the microtubules are here. And in dendrites and soma, which this is, they are interrupted in a mixed polarity. Microtubules can point one way or the other way. And in this situation, they're mixed. They're up and down, mixed together, and interconnected by these networks. So what I'm saying is that this is the most likely place for consciousness to occur in these mixed polarity networks of microtubules in pyramidal soma and dendrites in layer 5. Now, it could happen elsewhere, but if it's going to happen anywhere, it's going to happen there. And there's Bing.
Is that beige part down the bottom? Is that the?
This is the axon.
Axional.
That's the axon. Axon initiation segment, axon hillock. Yeah, it'd be there. This is the artistic interpretation to make it look nice, but yeah, this is the axon. This is the axon hillock or axon initiation segment, they call it now.
So is the Bing right above that? Or
I'm saying Bing is a collective effect, yeah, and also probably out here through microtubules there to other neurons. And here we see the structure of the microtubule. We'll come back to that. So, yeah. Now, a couple points. First of all, there's no pyramidal neurons in the cerebellum. Tononi makes a big deal about his phi complexity model predicting that there's no consciousness in cerebellum because it's modular or something like that. But there's no pyramidal neurons in cerebellum. So if you say, well, consciousness primarily happens in pyramidal neurons, then that explains that. End integration and cortical pyramidal neurons are the most likely site for consciousness from the previous summary slide. And microtubules in the pyramidal neurons are the most likely source of Hodgkin-Huxley deviation from several slides before. Therefore, microtubules and pyramidal dendrites soma are the most likely site for consciousness. So there they are again. And I want to make this point that if you record from a single pyramidal cell, and Christoph Koch did this work, from the soma and the apical dendrite at the same time, there's this perfect correlation, despite this being quite far away, 50 microns. Is that right? 50 microns away, they're perfectly correlated. And this can't happen by membrane propagation. And in the paper they say, well, it's because the noise and the background noise is coherent, is perfectly synchronized. But then what's synchronizing that? I don't think that's a really good explanation. I think it could be coming from inside, but we don't really know. The point is that there's this isopotentiality in these cortical neurons. And they don't like to talk about this anymore. It's like an embarrassment. So the final point about this is that the Bing consciousness might extend from here through interneurons and basilar dendrites in this layer, and perhaps even up to the cortical surface, since there's isopotentiality, and other neurons. Okay, so what about microtubules? Now, the party line is that they are the structural support, they're the skeleton of the cell, and they provide transport. So here we have a neuron. This is from this paper here in Science a few years ago. And this is the axon here, and this is a dendrite, because you see it's going to branch. So let's say you're synthesizing something in the cell body that has to go to this synapse out here. How does it get there? Well, it's carried by these things called kinesin going this way and then dynein going that way. And whatever the cargo is needs to be delivered to a certain synapse, whether it's here, 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 or wherever. And that means that it has to jump from one microtubule to another, because remember the dendritic microtubules are interrupted and mixed polarity. So it has to jump from one to another, and then it's going to come to this branch point. It's got to go, do I go right or do I go left? So how do the motor proteins know where to deliver their target? Imagine FedEx or something like that. What's their addressing system? Well, it turns out that tau protein, shown here in the red, which is a microtubule-associated protein, which is the protein that gets dislodged and hyperphosphorylated in Alzheimer's disease, is the traffic signal and tells the kinesin when to jump off and deliver its cargo. So the tau location on the microtubule is like traffic signals or instructions. So how does the tau know where to go and what to tell the kinesin? Well, the tau is binding at certain sites on the microtubule. So my argument is that the information is embedded in the microtubule that causes tau to bind at certain locations, and that's the coding for delivery of synaptic precursors for memory. And we know that if tau falls off, we get Alzheimer's disease. So Alzheimer's disease, all the beta amyloid outside the neuron gets all the attention and the money, but the real problem of the neurofibrillary tangles due to hyperphosphorylated tau and destabilized microtubules inside the neuron. The beta amyloid plaques by themselves don't really cause any problems unless they really invade the synapse, and even that's questionable. The problem comes from the what's happening inside the neuron, the cognitive deficit. The exact same thing happens in something called postoperative cognitive dysfunction in anesthesia. If you have too many anesthetics, cognition doesn't quite come back. It sometimes takes a long time. Sometimes it never comes back. Maybe we're unmasking Alzheimer's, but it looks like anesthetics, serial exposures to anesthetics destabilize microtubules and cause this tauopathy. So it's the same disease, pretty much. So what about memory? I mean, we still don't know how memory works because the membrane proteins, these things, are transient and interrupted, being recycled by those kinines and kinesins, dyneins and kinesins. So one of the things that happens in memory is calcium comes in and activates this calcium calmodulin, forming calcium calmodulin kinase 2, this hexagonal snowflake-shaped molecule, which then latches onto microtubules and distributes rapidly throughout the local network of microtubules in multiple neurons. And you can see that with immunofluorescence. Now, CAMK2, I might have talked about this last time, but CAMK2 is, you start with calcium calmodulin. Calcium comes in. So this is looking at it from the top, this is looking at it from the side. And it sprouts into this nano robot, or whatever you want to call it, I'm not even sure, with 6 legs, 6 kinase domains downward and 6 kinase domains upward. And here we see the kinase domains on the inner part of the feet, or hands, or whatever you want to call them. And these things phosphorylate something. And whatever they phosphorylate, since they're being flooded into the synapse, from the synapse into the postsynaptic area, dendrite or soma, whatever they phosphorylate is a good candidate to store memory, because then they're going to get recycled. So what could these things phosphorylate that would make sense in terms of running the show, in terms of organizing things and encoding memory? Well, we published this in 2012 that the geometry and size scale of the CAMK2 perfectly matches the microtubule hexagonal lattice. And we also showed in the paper that the phosphorylation gets down to the specific amino acids in the tubulin that can be changed and alter the state of the tubulin and encode information to bind tau or to do whatever, to affect vibrations, as we'll get to. So it could be that memory is encoded in microtubules. I think it's really the best bet, because membrane proteins don't last long enough. Neurofilaments are possible. They're very long-lasting. But at least for short-term or medium-term memory encoding, microtubules, I think, are the most logical candidate. And here is we show the capacity for information storage, depending on the A lattice, B lattice. humongous amount of information capacity in just one little neighborhood on a microtubule. So this is where memory could be. Which leads to the general topic of information processing in microtubules, which I first published on in 1982, wrote a book about in 1987, and then started applying Froehlich coherence and working with physicists like Steen Rasmussen, looking at quantum coherence, or not quantum, but coherence of microtubules processing information like microtubule automata. So the idea is that there's the synapse coming in, this is actin, and here's the microtubules. Notice how different they are in the dendrite than in the axon, where they're continuous and unipolar. So I'm saying the information, memory gets stored here, and processing goes on here, which then influences firing here. In other words, why are we paying attention only to the membrane instead of looking at what's inside? In medicine, it'd be like looking at dermatology instead of all of medicine, just looking at the surface. Okay, so another factor about this is, where do psychoactive drugs act to alter consciousness? Most people would say, for example, serotonin receptors, 5-HT receptors, the SSRIs, the antidepressants, they have this effect on the membrane, inhibiting reuptake of serotonin immediately, and yet it takes several weeks for the antidepressant effect to kick in because the microtubule cytoskeleton needs to reorganize. And I'll give you a reference for that later. GABA receptors are thought to mediate effects of benzodiazepines and anesthetics. I'll come to anesthetics later, but benzodiazepines actually don't really bind to GABA receptors. They alter slightly the binding of GABA to GABA receptors. The point is that all of these drugs are nonpolar, and they're going to get into the cell, and they're going to bind to microtubules also, in addition to anything they're binding to at the membrane surface, including opioids. Now, as far as NMDA, we'll come back to. So I'll just take these individually. I kind of said this before, but membrane effect immediate, but antidepressant effect takes weeks as cytoskeletal microtubules reorganize. There's the reference. GABA, they don't bind; they just alter the binding of GABA to the GABA receptor. And anyway, here's a GABA receptor, and here's the microtubule. So it's kind of like the GABA receptor is the tip of the iceberg of the microtubules anyway. So it's literally one big system. NMDA receptors, ketamine. Everybody's always wondered about how ketamine can do something to the NMDA receptor and yet and be an anesthetic and yet cause dissociation and hallucinations and people floating on the ceiling, and then they come back and they're perfectly fine. It's a great drug in anesthesia, by the way. And it turns out they also bind to microtubules. This is from studies looking for ketamine toxicity because it causes postoperative cognitive dysfunction. And it induces tau hyperphosphorylation in, that's a microtubule. It's kind of hard to see. It's kind of dark. But trust me, that's a microtubule. And it's all messed up if you give too much ketamine. So ketamine binds to microtubules as well as an NMDA receptor. Okay, now anesthetics. I'm an anesthesiologist. I make my living passing gas, as it were, putting people to sleep, waking them up. I've been doing it almost 40 years. And in my field, we still don't really know how anesthetics work, although it's looking like they're acting on microtubules, which is what you would want if you have a theory of consciousness based on microtubules. And they spare non-conscious functions. We do evoke potentials during anesthesia. Patient's unconscious, but their brain is receiving inputs. So where do anesthetics work? Well, going back over 100 years ago, these two guys, Meyer and Overton, showed that the potency of a bunch of gases, which are anesthetics, and they looked at different animals, mice, writing reflex, what concentration they would get back up and get off their back, and tadpoles when they would start to swim, all different animals, and they got the MAC, essentially the ED50, the mean alveolar concentration, the potency of the drug, and look for a whole bunch of physical parameters of what they would correlate, what the potency would correlate with, and they found that the potency over many, many orders of magnitude correlated with solubility in olive oil, of all things, gas partition coefficient. So methoxyfluorine is the most potent anesthetic. You only need 0.25%, halothane 0.75%, and then just go up and up. Nitrogen in many, many atmospheres. But it all correlates perfectly over many orders of magnitude with solubility in olive oil. Now what that means is if you ground up your body into different solubility parameters, polar, like blood, water, would be out here. Non-polar, fat, would be down here. And right in here, the aromatic rings, the aromatics, phenylalanine, benzene type rings, and certain amino acids. 3, 4, and 5 are where anesthetics bind, which is probably where consciousness comes from, down here, not from the polar parts of the body or the parts of the brain. So Rod Eckenhoff, who's a researcher at Penn, has studied anesthetics, and he didn't start with a bias like it must be microtubule. He started completely open and did a number of very interesting studies, proteomics and genomics. For example, halothane, an anesthetic, binds to 57 proteins in the human in cortex, 23 membrane proteins, and 34 soluble cytoplasmic proteins, including tubulin and microtubules. Genomic functions point to functions of microtubules. They also showed that exposure of primary cortical neurons to halothane alters genetic expression of 7 proteins, 3 common to both anesthetics, tubulin, and 2 others which don't seem to have anything to do with the signaling mechanism. No changes in membrane protein genetic expression. So this suggests that anesthetics are doing their functional action on microtubules. Proteomics and genomics point to microtubules. Another study they did that just came out earlier this year, direct modulation of microtubule stability contributes to anthracene general anesthesia, where they gave this is optogenetics. They gave this fluorescent anthracene to tadpoles who have conveniently transparent heads, and then they could illuminate them with the fluorescent wavelength and make the active anesthetic, and the tadpoles turned over. They became anesthetized. And then they ground up their brains, they donated their little brains to science, and they found that the anthracene was bound to microtubules, to tubulin. And they mentioned it was consistent with our quantum mobility theory. That was nice of them. So in tubulin, so here's a tubulin molecule. And Travis Craddock did this work, and he located the aromatic rings. Remember the nonpolar aromatic rings from amino acids tryptophan, phenylalanine, and tyrosine, where anesthetics bind, where consciousness is likely to come from, somewhere in the brain. And here we see in the red the halothane molecules in the midst of all these aromatic amino acids. There's actually 32 amino acids in tubulin. And here's where the anesthetics bind. They're very close to the aromatic rings in kind of a hydrophobic nonpolar region. It's like oil and water. The oily nonpolar groups want to get together and get rid of the water on the outside. So the water's out here, and inside it's convenient and conducive for anesthesia binding. So if we take a step back and say, okay, information processing in microtubules, we have about 1 billion tubulins per neuron, switching at about 10 to the 7th. I'll tell you where I get that number later. Ops per second, it's 10 megahertz. Gives about 10 to the 16th operations per second per neuron. Now the singularity and all those guys are saying there's 10 to the 16th operations per brain per second. So they don't like this very much because it pushes their goalposts way down, way into the future. So I'm not very popular with those guys. Nor is Roger, for different reasons. Roger Penrose, my collaborator. This gives about 10 to the 27th operations per second for the brain. But this doesn't explain consciousness, feelings, awareness, experience, nor understanding. Roger Penrose's Gödel theorem argument can boil down to the fact that computers can do a lot of amazing things. They don't understand anything. And John Searle made that same point with this Chinese room. It's pretty much the same argument. Is consciousness a computation? I don't think it is. In fact, I'm coming around to the point of view that consciousness is more like music than a computation. I'm not quite sure what that means, but it's more like music. I'll just leave it at that. So where's the Bing? How do we get there? And so a little bit about the quantum physics. Roger wrote this book, and I've been studying microtubules for years, but in the early 90s, but didn't have a mechanism. It had a structure, but not a mechanism. He had a mechanism, not a structure. He basically said that quantum superposition is, I went over this last time, so I'm going to go through this quickly. Separation in underlying reality, like the multiple worlds. The separations are unstable and reduced by an objective threshold given by the indeterminacy principle. Each such OR event is accompanied by subjective experience, and the choices are influenced by Platonic values embedded in space-time geometry. So he did a very nice cartoon where matter is curvature in space-time, so a particle in 2 different positions would be curvature in slightly different directions. A superposition of the particle being in 2 places at once would be a separation in the fine-scale structure of the universe, like the multiple worlds hypothesis, except that these separations are unstable and after time t will self-collapse, and that's a moment of consciousness. I won't go into the rationale. Several books written about this, and it's kind of like Sherlock Holmes said, when you rule out the impossible, whatever's left must be correct, no matter how seemingly improbable. That's one way to look at it. But there's more to it than that. But I mean, Christophe's now a panpsychist, so if consciousness is a property of matter, I think it's more likely that it's a property of wave function collapse. Now, the Copenhagen interpretation of quantum mechanics of the consciousness causes collapse. This is saying that collapse is consciousness. When you have this type of collapse, by the indeterminacy principle, you have a little moment of consciousness. But they're very simple normally. They have to be organized or orchestrated to make full consciousness. These are ubiquitous. They're the same as decoherence, but the subjectivity lacks cognition or meaning. They're proto-conscious moments, but they're discrete snapshots, kind of like Christophe said in that article, like whitehead occasions or Buddhist moments. So I think a lot of converging ideas give us that consciousness is a sequence of discrete events. And this is consistent with Eastern thought that proto-consciousness is everywhere. But how could these be orchestrated, occur in the brain and be combined or orchestrated into meaningful cognitive fully conscious events with rich phenomenology? Microtubules are the answer, acting as quantum computers. So we developed this theory, the orchestrated objective reduction theory, with a protein qubit and microtubules inside neurons. And first started in 1994, this was. There's Dave Chalmers on the far left, Roger in the yellow shirt. And the basic idea was in a microtubule you had a buildup of quantum superposition that reached threshold for collapse by equals h over t. There's a moment of consciousness, and these selected tubulins then go on to trigger firing or not trigger firing, or store memory. So the basic idea, original idea was, if you go back to this space-time qubit or quantum bit, we equated it to a quantum bit of a tubulin being in two different conformations and a superposition of both to build a quantum computer. It turns out you don't need this conformational change, and it causes trouble because it requires energy and generates heat. So in 2002, we switched to a more physiological, sensible idea that there's really dipole states along these, remember the aromatic rings can have dipole states in different directions, and a superposition of dipoles in both states. So I showed you this before, and this is very similar to what's happening in quantum coherence in photosynthesis proteins by exciton hopping. So in photosynthesis, plants collect photons and transfer the energy to make food through these multiple chromophores, which are essentially acting like the aromatic amino acids do, we claim, in tubulin, to get the energy over here. And the excitons occupy all possible pathways simultaneously to get there, which is why we have efficient food production while we're all alive today. Without it, we might not have life on this planet without very efficient photosynthesis due to this quantum coherence mechanism. And I think Bridget Whaley is here, maybe Fleming is too, here. Some of these guys are here at Berkeley. So basically, the idea is that we have a pathway, which is where anesthetics act, that give us the dipoles, and these dipoles are representing the information, and to make a qubit out of a dipole pathway qubit. Now, it also could be spin. In our recent paper, Roger and I said it also could be spin in different directions, pretty much the same idea. So we have topological bits or qubits, which could be vibrational states of the microtubule. So the idea is that the dipoles, different states, we have a quantum superposition, which I think I can talk about this later if anybody wants to discuss further. Psychedelic drugs may actually promote this quantum superposition and increase the topological vibrations in the microtubule. So, okay, that's all theory. Now, what about evidence? So this fellow, Anirban Bandyapati, from National Institute of Material Sciences in Tsukuba, Japan, is now at MIT. And in 2009, he began to do these studies of an individual microtubule, and he put 4 electrodes on an individual microtubule, 2 to stimulate and 2 to record. And if you don't stimulate them, they're conductors. But if you stimulate and sweep the frequency of stimulation, you find resonant frequencies. For example, at about 10 megahertz, 20 megahertz, down as low as 10 kilohertz. you can see the microtubule at the top. So at these particular frequencies, the resistance drops. Now it's not quantum necessarily, because you have a finite resistance between the electrode and the microtubule, but you get these sharp resonance peaks at specific resonant frequencies in megahertz, and there's a whole bunch of them. So this is an individual microtubules. You can show here's the resonance peaks here from about 10 kilohertz through megahertz through gigahertz, actually, in individual microtubules. He then, I forgot I got to show you this. Well, I'll come back to that. So he took these scanning tunneling microscope tips. So this is nanotechnology, where you can see atoms. You move with piezoelectrodes very slightly, and basically electrodes normally used in physiology are humongous compared to this, which comes to a single atom tip. So you can insert it, say, up to here into the neuron, and it doesn't perturb the neuron. You get much sharper signals from what's going on inside the neuron. So he then, for example, could put 2 of these into a single neuron process, into one dendrite here, for example. And here's one that's actually broken. At the very top, that bridge is just a bundle of microtubules and actually recorded right from that, so you don't need the membrane. And, okay, that's what I want to show you. So forget all this other stuff. Okay, so he did those studies, putting them into neurons. And these were from individual microtubules, and then he got, this is from from microtubules inside active neurons. These are in culture while the neurons are firing, the membranes are depolarizing. And the caveat here is this hasn't been published yet. It's being written up by the people at MIT, and I'm waiting to hear the status of that paper. So the point is that you have, in this case, 4 resonance bands that are spaced about every 3 orders of magnitude, kind of like a fractal hierarchy or something repeating over scales. Now, what's missing down here? Well, I'll come back to that point. So by theoretical predictions, let me go back. So we have 10 to the third, 10 kilohertz, which gives 10 to the minus 4th seconds coherence time. By prediction and experimental evidence, we have coherence times up to 10 to the minus 4th seconds, but they're still too short for known correlates of consciousness like EEG, 40 hertz gamma synchrony, and so forth. So Roger came up with the idea that there were like inverse harmonics or beat frequencies. So if you have like megahertz vibrations, they're slightly different. They're going to give beats, just like in music. And here we have like 800 versus 840. It's too hard to make this megahertz. And you can see that you get beats at 40 hertz. So in our most recent paper, Roger Penrose and I suggest that EEG is the tip of an iceberg of a fractal hierarchy of dynamics that goes into kilohertz, megahertz, gigahertz, maybe faster inside the neuron in the microtubules. And that what we see as EEG is the tip of an iceberg of this fractal hierarchy. So here I've taken the previous slide and I've just put in the EEG spectrum. And you could imagine it would go negative like these, but it's not how it's recorded. So this is just the EEG spectrum. You see it fits fairly well as a 5th resonance band of what's going on. So I think activity relevant to consciousness goes from gigahertz, megahertz, kilohertz, up into hertz in EEG. So consciousness occurs across scale, and as I said before, it's more like music than computation. And if megahertz quantum vibrations and microtubules mediate consciousness, then applying megahertz vibrations, for example, ultrasound, to the brain should affect mood. And we heard Sterling talking about that before. So I was talking about this with my colleagues, and I said, we should really try this on chronic pain patients who are depressed, see if it affects their mood. And they said, we're trying on ourselves first. It's your idea. You got a shaved head. You're first. So there I was. And actually, I tried it myself first. This is a follow-up. And I gave it to I put it here for about 15. This is an ultrasound machine we use in the OR. And it gives imaging, because what happens, the waves go in, they bounce back, and this recedes. And so the thick line is my skull, and you can see the layers of my frontal and temporal cortex. It's a lousy imaging device because it's got to go through the skull twice to get the picture. But it's approved for imaging, which is how we got it approved to the IRB, because it was already approved for brain imaging. So as Sterling mentioned, recently Jamie Tyler's group published in Nature Neuroscience that focused ultrasound improves cognitive discrimination and sensory discrimination task. They're using focus. They're trying to aim at one point. The device I use is scans. It's kind of like a broad spectrum hitting wide parts of frontal and temporal cortex. You can also focus it. In fact, ultrasound, unlike transcranial magnetic and transcranial electrical, can be focused to depth. So we published the first study on mental states, mood, showed a slight improvement in mood. Then I got my colleagues involved, Jay Sanguinetti, and we have several studies done that haven't been written up yet, improves mood in volunteers. And then 2 other studies at the cellular level, Uma Raman, who presented the Consciousness Conference, and Sadviki Gupta, and I'll show you what they showed. So can you see these?
We could kill the lights.
Yeah. All right. So, thank you. So, Uma Raman, is an undergraduate student, did this in Saurav Ghosh's lab, and she got cortical neurons, embryonic neurons, grew them in culture, and before they formed networks, exposed them to ultrasound for, I think, 2 minutes, 2 megahertz for 2 minutes, I think. I'm not sure. Anyway, and then 4 hours later, she counted them all, blinded as to which ones got ultrasound and which ones didn't, and just counted how many in each group had more neurite, because these things eventually will grow neurites, like here and here. You can see that. And anyway, she got a significant increase in the number of ultrasound-exposed neurons that had neurites. And by 24 hours, this is at 4 hours, by 24 hours, everything had averaged out from the one exposure. So it probably needs to be repeated. But in a brain injury, traumatic brain injury, since you want to make new connections, you want to grow new, not necessarily new neurons, but new connections from damaged neurons, stimulating, assuming it's working on microtubules, whatever it's doing, if you're making new neurites, that should help. And also because of Alzheimer's disease, the microtubules fall apart. It should help that also, maybe. Now, about Alzheimer's, Sadviki Gupta did a study on polymerization, microtubule assembly, and with and without ultrasound. And you can't do it in the device because you can't put the ultrasound in the spectrophotometer where you measure. But they figured out a way to do it, and then make a long and then checked afterwards, and the sample with the ultrasound exposure stayed fairly constant, whereas the sample without ultrasound exposure started to decay or to depolymerize, suggesting that the ultrasound stabilized the microtubules and promoted microtubules as opposed to disassembling. So this is why it should, in principle, be helpful in Alzheimer's, because your microtubules fall apart. And we're combining the last 2 studies into one paper. I'm going to write up. Okay, so I'm going to close and just say here are the references from the recent paper I wrote with Roger Penrose, including a reply to 2 different groups of commentaries, including the bottom one, which was quite negative, and we had to deal with them separately. And so let me conclude by saying that quantum vibrations, or just vibrations, orchestrated reduction microtubules mediate consciousness, regulate neuronal firings, and connect consciousness to the fine-scale non-local structure of the universe, which is how you solve the hard problem, I think. EEG and neuronal membrane activities may emerge as beats of smaller-scale, higher-frequency microtubule vibrations, for example, 10 megahertz. Consciousness extends over scales, like music. Tuning the brain by treating quantum vibrations, brain microtubules via megahertz, ultrasound, TUS, drugs, because I think drugs are acting on microtubules anyway, even though we don't realize it, or meditative techniques where you're chanting and doing resonances in your body may promote this, may activate microtubules to improve mental and neurological conditions. Thank you.
When you say microtubules are in like a quantum superposition of states, do you know like how robust those superpositions are to noise? Because I know from like a physics background, it's really, really hard to
Right.
get something into
Decoherence, yeah.
a superposition state. So do you have any idea of like what the time scales are?
Well, we know that quantum coherence time is 10 to the minus 4 seconds from honor bonds, so we know it's at least a 10,000th of a second. So in 2000, Max Tegmark published a paper that was kind of an ambush. I think he was out to get Rogers, kind of like a Wild West gunslinger shooting for Wyatt Earp or something. So he wrote this really stupid paper in Physics Review E that said that, and the surface attempted to prove the obvious. The microtubules are too warm, wet, and noisy for quantum coherence. And he came up with this, so here's his decoherence formula. The coherence time is going to be equal to, notice the temperatures in the numerator, which doesn't make sense, because as the temperature goes up, this should lengthen. But he calculated 10 to the minus 13 seconds decoherence time. Way, way, way too short. However, he made a couple mistakes. The superposition separation he used was 24 nanometers, whereas our separation is the Fermi level, femtometers, the level of the nuclei. So a superposition, a tubular superposition means that it's separated from itself by the diameter of its atomic nucleus, which is extremely tiny. That's number one. Number two, it's happening in the quantum. So when you correct all this, you get to 10 to the minus 4th seconds, which has been shown experimentally. It's also happening in the nonpolar hydrophobic region, away from the polar thermal interactions. So everybody says the brain is too warm, wet, and noisy. It's not too warm because we now know that there's quantum coherence at room temperature, Bose-Einstein condensates, photosynthesis. It's not too wet because it's happening in the nonpolar dry part of the cell and of the protein where there's no water, there's no polarity. And it's not too noisy because the noise in the brain is actually correlated and it's probably signal. So what you just said was the party line, and is the party line, but it's just been shown to be wrong. That there's warm, otherwise there wouldn't be photosynthesis happening in sunlight, for example. In the back, Dr. Searle, John Searle.
Well, I have so many questions. I don't know where to start, but let me start with one I'm sure you've heard before, and that is what's special about neurons on your account. That's one superposition. That's nothing special with neurons.
Only in neurons are only in neuronal dendrites and soma, not even axons. Just neuronal dendrites and soma are microtubules interrupted. Okay, all other cells, they start in the cell body, near the nucleus cell body, and go like spokes of a wheel continuously. They're not interrupted, with a plus inward and the minus out. They're all the same polarity. In dendrites and soma, they're interrupted. So obviously they're not there for structural support. You wouldn't want a girder in a building to be broken and then start over here. They're short interrupted and of mixed polarity. It's unique in all of biology. No other cell, and even in neurons, only in dendrites and soma. And they can be, I used to think they need to be entangled through gap junctions. That may or may not be true because Anirban showed they can be entangled across neurons. So the uniqueness comes in the dendrites and soma of brain neurons.
Is this specifically pyramidal neurons?
No, all dendrites and soma, even peripheral ones, I think.
So
But
the neurons in the stomach could be conscious, for example.
some people, yeah, or the heart. Some people argue for the heart, but you know, it's possible. There could be memory in the heart. I mean, we hear all these weird stories of people get heart transplants and pick up memories from the donor, and I don't know if that's true or not. But I think only in the brain do you really have enough of these connected in one coherent system to have consciousness.
What about water? Coherent water in the matrix outside?
Yeah. Outside the neurons?
Yeah.
Yeah, I don't know. Maybe. Well, I think there is non-locality, whether that needs to go through water or not. Some people think the water inside the microtubule hollow core is ordered in a coherent state in gigahertz. So there's all kinds of possibilities with water. But in
The work of Gerald Pollack
Yeah.
in that fourth phase of water, and the surface areas around all of the materials in the brain, there's going to be this extended zone, he calls it.
Right. No, I'm all for that. I know his work and I like it a lot. And I think that that's going to be more conducive to avoiding decoherence if the water is ordered, if they're all vibrating at the same frequency, for example. So I'm all for that.
Well, Walter Freeman has been suggesting basically a quantum field theory with Vitiello.
Right.
And, you know, his approach has basically come to that as a conclusion. And it's not at all incongruent with
Right.
what you have, which is really brilliant in a way.
Well, thank you.
Because you have an extended condensed phase. You have to explain all parts of the condensed phase,
Right.
not just one part.
Yeah, I know that work. In fact, I wrote a commentary on their paper about dark energy or something,
Yeah.
and they basically had two fields, kind of kind of like the integration field and the firing field.
Right. It's really a question of what is the appropriate mathematics to treat it. I'm not sure what org theory is, but it seems that there's a rich apparatus for QFT that would be more appropriate to use.
Maybe, but it comes down to almost a religious preference. For one thing, QFT doesn't have collapse,
It
and
doesn't come down to a religious preference. There's a coherence and history in condensed matter physics.
right.
It's not like the history is there.
Right.
It's been validated for.
No, there's no problem. I have no problem with quantum field theory, but there's no collapse in quantum field theory. And being
Those phase transitions that
well, you could say that.
can be integrated, and I think Vitiello and others have been talking about that. Vitiello had a fractal analysis of phase and time just 3 years ago.
Yeah. And he's also talked about microtubules and dissipative dynamics. And I just saw him at the Tucson conference. No, I'm all for that. I guess the only difference, the difference would be, we're pinpointing in the microtubules in these quantum channels, we're calling them, inside the microtubules. With the pathways I showed you would correlate with specific vibrations. So, for example, around one helical pattern would be 10 megahertz, another one would be something else. So the pathways can correlate, which means that experimentally what we want to do, and I hope to do this next year or two, or hope Honorbound does it, is give anesthesia to these neurons with the intraneuronal scanning tunneling probe to see if the high-frequency vibrations go away, and also to give psychoactives. I'd be very curious to see what happens if you gave LSD to one of these neurons. I would bet that the frequency would go up, that the resonant frequency would shift maybe towards the gigahertz, for example. So
Can I ask?
Yeah.
Dick Presti is sort of perhaps knowledgeable. Is there a cell-based model for psychoactive drugs which could be directly employed to do that?
Well, I think the idea that internal binding of various drugs to microtubules is a great, wonderful and eminently testable hypothesis. So more power to
Yeah.
you for
But
doing
is there a
that.
cell-based asset?
Well, that's a very good question.
There is.
I talked. Well, I think there is now. There didn't used to be. I got started on this end of it. I was talking to one of our chair of pharmacology gave a talk about new opiates, non-addictive opiates, which I thought was a pipe dream, so to speak. But and because this particular drug was it caused despondency. I said, well, you're worried it's going to, it didn't cause euphoria, but it looked like it might cause. I said, you're going to give that to a patient? And go. Anyway, I said, it's nonpolar, right? He says, so it gets in the neuron, right? I go, how do you know it binds to tubulin? He goes, well, everything binds to tubulin. I go, well, why don't you study that? Well, nobody studies that. But then he had a very good point. He said, well, what are you going to study? Because up till recently, you would give a drug or something and see if the microtubules destabilized, stabilized, polymerized, depolymerized. That's all you could look at. Now, we can give drugs and look at these vibrational frequencies of the microtubules and see what happens to them. Like I'm saying, I'm predicting that psychoactive drugs will shift it to the high frequency, anesthesia will take it away.
This is really fascinating. What is your take on oncogenesis? And we'll
Maybe we could take more questions and then.
I do have a take on that, but Sterling.
I just recently had a family member pass away, and I was wondering, how does your model account for brain death, like lack of oxygen, and is it in the case of brain death? Is there a way to possibly freeze the brain and take it layer by layer and then recreate it by measuring the states of the microtubules? Or is that even possible?
Well, you raised about 6 interesting subjects there.
Yeah.
Let me first say that end-of-life brain activity. We use these monitors in anesthesia. They're simple EEG devices. They're just frontal because nobody wants to mess with anything else. And they process it, and they give you an idiot number from 0 to 100. It's something to do with EEG, probably gamma, probably temporal muscle. They don't really. It's proprietary. They give you an idiot number from 0 to 100. Awake people are 80 to 100. You want your patients between 40 and 60 so they won't be awake and they won't remember and they won't see you. That's basically what these things are for, because people waking up under anesthesia. So a few years ago, a palliative medicine guy at George Washington, Lakhmir Chawla, started putting these brain monitors on his patients as they were dying. You know, they decided to withdraw support, they were hopeless, whatever. So what happens is the number goes down. It probably starts, you know, below around 40 or lower because they're not really with it. It goes down to about 0, and then about the time the heart stops, there's this burst of activity that lasts 90 seconds to 20 minutes in some cases. And they analyzed it, and it was gamma synchrony. Now, George Mashour did the same study in rats that he presented at the Tucson conference, where he gave put all these fancy EEG monitors on rats, gave potassium to the heart, stopped immediately, and with 30 seconds, there's this burst of gamma activity. Now, what that means, nobody knows. But it's not random spasmodic firings or activity of neurons because it's highly coherent across both hemispheres. It sure looks like consciousness as far as anybody. Now, is it just because it's this huge stimulus? Hey, I just died, and now what? Or some people suggest it could be the soul leaving the body. Well, who knows? I mean, you can't. If consciousness is happening in space-time geometry, it could be non-local. We can't. You can't say it's impossible until somebody can show how consciousness occurs in the brain. You can't say it's impossible out of the brain. I'm not claiming any proof for that. I'm just saying you can't say it's impossible.
Did the MIT microtubule studies did they find instantaneous instantaneous transport along the tubule with the?
Then we're looking at transport. They're looking at just individual microtubules and measuring the conductance between two electrodes.
Yeah, I'm just saying. So, but if there's a conductance between the two electrodes, then that means that it's not synchronous entirely.
Yeah, it's a very low. It's attawatts. It's some really low power. What you're saying, is there a flow, or is it?
I mean, like, so you were talking about how the neurons themselves are, like, they down the axon you have some sort of coherence.
Yeah, or dendrite.
Dendrite, you have some sort of coherence, and you would expect to find the same thing along the microtubule. If that.
I'm not sure about that. I see what you're getting at. They're applying AC, and they're getting. It just puts it in a state. You're saying that there must be flow along that. But it might be just one electron moving a little bit and shoving the next one down. So it's pretty much, for all practical purposes, it would be instantaneous. And then we know that happens in neurons through the isopotentiality. Right. That's a good question. I have to think about that. I think I see your point.
You had said earlier from the Christoph paper that they believe that consciousness happens in the third pathway, right, from the frontal cortex.
Well, that was before all this stuff, but I think he deserves credit for presaging that.
Okay, but you would argue against that consciousness is happening through all those things?
No, I think it's in the third wave. I think if you think about, you need time to build up to reach threshold for self-collapse. It's an interesting question, but I think it's probably happening in the third wave.
Because those microtubules and those neurons are present the whole time.
Right, as they are in every other cell. So why aren't they happening in one or two? I don't know. That's a good question. I think for some reason, maybe you need to get to the pyramidal cells. Maybe you need the...
pyramidal cells in B1, right? Like early visual cortex, there's pyramidal cells.
Yeah.
And they're constantly receiving input. So why isn't there consciousness there? Why does it have to go to prefrontal cortex first?
Well, I don't know the answer. That's a very good question. I've thought about that. But by E equals H over T, you need a significant amount of time. And or even if it's B frequency, so you need enough time to reach threshold for self-collapse. But that's a good question. I don't have a good answer for it. Okay. I'm thinking about it.
Time is kind of continuous, right? Like we're constantly receiving. What is that time with respect to? Like enough time from what? From onset of stimulus, or?
All right. So if you see me, and by the time it goes to the back of your brain, the front of your brain, and then projects to the third area, that's probably going to be 300 to 500 milliseconds. But yet you may respond to me at 100 milliseconds, which is taken to mean that your consciousness is epiphenomenal and illusory. The only way around that is to have this funny backward time effect that Benjamin Libet discovered in 1979 that's been rediscovered over and over again, but which the neuroscientists, including Christoph, who found it in his own study but discarded it, this funny backward time effect, which can give you, send information backward in time so that you can respond consciously in real time. So even though the activity that tells you what to say hasn't happened yet, it's sent backwards in time. Now, this backward time effect can only happen in quantum mechanics, quantum physics, and it could happen in the brain. I have another paper about that called How Quantum Brain Biology Can Rescue Conscious Free Will, which shows how backward time effects is necessary for real-time conscious control.
I don't think you answered his question. The question is, if the microtubules in V1 are firing continuously, then there's a continuous passage of time right there in V1. Why isn't it conscious?
I don't know. Okay, I think you could say that they don't have the right conditions, they don't have the isolation, they don't have the right configuration, but I don't know. I think it has something to do with time, but I don't have a good answer for that.
Cell types across the neocortex are quite similar, right? Even in V1, and then if you go to prefrontal cortex, the neocortex is very uniform across, right? So there's different regions that may respond differently or act differently or do different things, but the structure of a hypercolumn is about the same.
An answer to that question would be it is conscious, it's just not us.
That is an answer.
Yes.
So you sound like a higher order thought person, that it has to go to prefrontal cortex for it to be us, to have sense of self. It could be.
Well, like Tononi has complexes, and the main complexes are experience, and then subcomplexes,
A zombie.
for example. Not that I believe Tononi.
That's good.
I was going to say about the Tononi point. Tononi has this theory that there's like some qualia space of consciousness and some sort of geometric shape, and how you can measure integrated information, and that's a measure of consciousness in some way. And so, does your Platonic values at space-time geometry, could that somehow relate to Tononi's theory on building some integrated information? And
Yeah, maybe, except
higher in prefrontal.
space-time geometry, in my view, is something real. It actually exists at the Planck scale. And the other thing about that is somebody pointed out to me that Tononi's, his whole thing is based on phi is from the golden mean and the Fibonacci series and all that geometry, but he doesn't have anything physical to hang it on. It's all in, I don't know, Hilbert space or abstract space or something. Whereas microtubules have Fibonacci geometry and the hexagonal geometry and all the other stuff that he's talking about, but he doesn't have any. He has nothing to hang it on. That's what I'm saying. I'm hanging it on microtubules and then space-time geometry. Other than that, exactly the same, except he doesn't have any biology or philosophy in it or physics. Yeah.
The Penrose's idea of the beats, the local field potential recurring to the beats. The beats would be very, the frequency of the beats would, just from physics, would vary quite a bit if the actual parent frequencies varied a tiny amount. And it seems like...
Well, excuse me, it's the difference between the two.
Exactly.
So if they change the beats are going to stay the same. But EEG does change.
If they change together, well, that's the point. It seems like the EEG would smear out quite a bit if the higher frequencies vary just a very tiny amount in their, because they're so high. Unless they were totally in synchrony, the resulting beat frequency would change quite a bit, and I don't know that we see that. I mean,
Well, gamma comes and goes in different areas.
yes, but it doesn't seem like the numbers would work out because
Well, the numbers in our paper.
Unless you're talking about a high synchrony between the higher frequencies that would change to produce those beats.
Well, I think I think there can be fairly constant synchrony. Microtubules structurally have this resonant structure, and at a given amount of energy, they're going to vibrate coherently, synchronously. So I'm not sure I see that as a big problem. What you're saying is if this one changes and this one doesn't,
Yes.
yeah. Well, yeah, maybe that's true, and the EEG would go away for that particular area, or change, or slow down, or whatever. But if they move together, then it should be okay.
Or gamma would just move,
Yeah.
down into beta. or
That sounds like Gautam's study of where you see the patterns moving. I meant to say that during the looking at the pyramidal cells, and this also might relate to John's question, why you're not conscious in V1, that it needs to be within some kind of envelope that can move around. But I'm not sure that's the right answer. John?
Still, they seem to have a question, and that is, if consciousness really is a property of individual microtubules in the third phase, and presumably billions of them, why is there a unified conscious field? Why does my consciousness come as a single conscious field?
Entanglement. Because all those microtubules are entangled into one unified, just like a Bose-Einstein condensate. And it was just shown that Bose-Einstein condensation, something like what a laser is, can occur at room temperature. It used to be at absolute zero temperature, but now they're doing it at warm temperature. So your consciousness is entangled as a unified, coherent entity. Maybe there's consciousness, like you said, in V1, but it's not entangled, or something like that. Actually, I kind of like that idea, because you could call it proto-conscious or something like that. But binding or unity of your consciousness is because all those microtubules, or portions of a bunch of microtubules, are entangled and coherent in one system.
So does that entanglement, so you have this coherent time period, right? That you were talking about with regard to Tedgemark, for example.
Oh, yeah. So we know...
The entanglement, does that have to occur?
During that time, it's entangled, yeah.
And so all of them are lasting that time moment of how many?
Well, we know experimentally that 10 to the minus 4 seconds at warm temperature, because we have 10 kilohertz resonant frequencies.
So somehow during that time period, they're all entangled.
Correct. And then at the end of that interval, at the end of 10 to the minus 4 seconds, there's a collapse. But that's too fast for us to recognize. But if we bring in the interference to give the beat frequencies, so we have epochs that are much slower.
So normally entanglement is induced by, it's like some common source of interaction.
Yeah.
And what would that be in this case?
Well, you can do it with a laser, or I asked Roger this once. He says, well, the natural tendency for things is to be entangled. The question is, why aren't they entangled? And he has this thing about quanglement. So the natural tendency, but if you want to make an entanglement system, you have two systems and you hit them both with the same laser and they become entangled. Something like that. The microtubules are acting a little bit like lasers, according to what Froelich said, 40 years ago, 50 years ago.
All right, we will let the audience free, but feel free to chat with Stuart now and play around with this ultrasound device. And thank you for coming.
Thank you.